So my talk today this afternoon is on anchored multiplex PCR for targeted next-generation sequencing. Give some of my disclosures. Stephen already mentioned I'm a co-founder of ArcherDX, which has been acquired. I remain as an equity holder of Enzymatics as well as consultant for Enzymatics. I am medical director of the CRISP lab at the Broad Institute. And also my wife is 36 weeks pregnant, so if I have to dart in the middle of my talk, you'll know why. So the layout for my talk is that I'll make a case for broad-based cancer genotyping at the clinical setting, describe our approach at MGH, and also talk to you about how we are approaching next-generation sequencing at MGH, and in particular how we have implemented the AMP technology. So I list this here as a new paradigm in terms of cancer treatment, but really it's becoming the mainstay. In multiple medical centers across the United States, this is actually being practiced day to day now, is that rather than just simply taking a tumor resection and going straight to chemo or radiation therapy, you take a detour to the molecular pathology lab, find something, a molecular defect that would actually define the disease in a more specific manner, and then drive the patient in terms of a targeted therapy in hopes of achieving better and safer response for the patient. There are a number of examples of this going on today. 4 are listed here: EGFR in lung cancer, BRAF, V600 in melanoma, crizotinib, and soon ROS, if not already, ROS1 translocation in lung adenocarcinoma as well. And then you have your KRAS mutations in terms of resistance to cetuximab. I'm proud to say that actually 3 of these came from the MGH as a story in some form or fashion. And like I already mentioned, ROS1 is another one that's coming out from MGH as well. So this is the type of response that you can achieve if you have the right therapy combined with the right diagnostics tool and have sort of a perfect setting for all of this to come together. So this is one of the first 2 patients who was put on crizotinib, an ALK translocated lung adenocarcinoma patient who, upon treatment 2 weeks later, and this is an oral compound, you can see a dramatic response in terms of the PET-avid image here. You see her primary tumor as well as METs here, and within 2 weeks, essentially the disease is gone, if not a little left there. The story is very interesting because it defines something that is sort of unheard of in the field of drug discovery and biopharmaceutical implementation in terms of clinical management of lung cancer patients. The drug was founded in 2005. Internally, Pfizer had already shown some feasibility in terms of response to some cell lines with this drug. And I believe internally they didn't know that this was an ALK translocated case. At 2007 or so, mid-year, the Japanese group who discovered this in lung cancer patients published their paper. And quickly, John Iafredi, out of his own madness or whatever, took the ALCL VISIS FISH probe, converted that into a clinical assay for lung cancer, and applied it on FFP material as a routine clinical test. John also had the knowledge, working with Jeff Settleman at MGH, that this particular drug, there were 2 cell lines that responded to this drug in vitro dramatically, and they happened to have the ALK translocation proved by the FISH testing that John set up. It wasn't long before we found 1, 2, if not 3 or 4 patients. And serendipitously, at the same time at MGH, there was an open phase 1 trial for this particular drug, not for ALK and not for lung, but rather for MET amplification in esophageal adenocarcinoma. That trial never came to fruition because these patients were dying very quickly, and the incidence of MET amplification in esophageal adenocarcinoma was well below 1%. However, when they funneled this into this open trial, those 1 or 2 ALK positive patients, this was the effect that they saw. So really in this case, you don't need an N of 100 to believe this. You just need an N of 1, N of 2, and to see a dramatic response like this, given a molecular defect and signature in this setting, is clear-cut positive proof that you have something real going on. And before long, we had 10 patients, 20 patients, 50 patients, all showing similar types of responses. And we have the story, which is called ALK translocation responding to crizotinib in lung adenocarcinoma. Fast forward 1.5 to 2 years later, this drug is approved by FDA for lung adenocarcinoma with the translocation, and as well the companion diagnostic, which is the FISH VISIS probe for ALK translocation, was also approved by the FDA for testing in the clinical setting. So you can see that from effect, observing this in vitro, to patient implementation and management really took no more than 2 or 3 years. And this is unheard of. And this, I think, is how cancer treatment will be practiced from here on out, if you marry the right testing with the right patients and the right drug. So that same study that I mentioned here showed those 2 cell lines, non-small cell lung carcinoma here and here, which responded to the drug. But it happens to be that there was a 3rd cell line here, HCC78, which also responded moderately to the drug. And you know what? This is the ROS translocation case. Again, John developed another ROS translocation assay using FISH probes, funneled the first patient through, same story. Crizotinib response to the lung cancer response to the drug, and it's because of the signature of ROS translocation. So these 2 stories, probably in the field of oncology in the last 2 years, are likely 2 of the biggest stories. Other than what you've heard about BRAF and also CML, BCR-ABL, there's no other stories like this. And this can happen, and it's real. It's being done today, and it's just in the last 2-3 years that it has come to fruition like this. But we realize that to be able to do more of these types of incidental sort of cool findings and to be able to change cancer management, you really need a clinical genotyping platform that can really screen patients in real time and be able to funnel them through these types of clinical trials to do first in human, if not different sort of ancillary types of treatment for patients and be able to see these responses. So here I'm listing some of the criteria that I think is crucial as part of running a CLIA or a CAP lab that you have to have in place in order to achieve this. Obviously, it has to work well on formalin-fixed paraffin-embedded tissue, have good turnaround time so that you can meet the demands of the clinicians. It needs to be actionable and all of these other sort of logistic things, which I can't cover in full today. So if you move back 4 years ago, this task was very easy. At the time, next-generation sequencing was immature. Error rates were high, difficult for a small lab, particularly like us, to run a HiSeq or one of the Illumina instruments back then. But we knew that there were low-hanging fruits already. If you just look at COSMIC at that time, there were these hotspot mutations like KRAS, where if you just combine this into a panel, you'll have some successful type of assay that you can launch as a clinical test. And that's what we did. So we selected the snapshot genotyping-based assay, which is essentially a multiplex PCR, 7 or 8 per tube, following that with single base extension genotyping with varying lengths of primers here, and using capillary as the readout. So we did this across 70 or 80 point mutations, launched the assay, and it has worked pretty well. Even today, we're using this assay. And this is just showing you our experience with lung cancers, a subset of what we've tested so far. Because right now, we're likely up to 1,500, 2,000 cases in the last 4 years or so. What you can see is that for about 60% of cases, we're able to identify some driver mutation. Quite a bit of them are actually quite actionable in terms of therapy. There's still about 40% or more that we don't know about, but we've come quickly to realize that cancer is more than just point mutations, and that's the limitation of a snapshot. It's also copy number, indels, and also rearrangements that are highly, highly important for you to detect in terms of cancer testing. So the other story here is that just in our small subset, we realize that there's this sort of tail end of mutations here. The days of BRAF, KRAS, and EGFR are likely over. No more are you going to find these sort of high peaks and mountains. More than likely, it's going to be about these low liars here, like ALK, like ROS, which are only about 2 or 3% prevalent in the lung adenocarcinoma cases. And this is recapitulated yet again in TCGA projects where if you look at melanoma, lung cancer, or any other projects that have produced data based on next-generation sequencing, you always get this tail end here. So the fact of the matter is, in this day and age today, if you're running a clinical molecular diagnostics lab for oncology, you have to be able to do multiple genes, not just one-off single-base gene testing anymore. And this is also highlighted by this real-world example. This is a requisition from one of our clinicians. It's sort of a roadmap of what to do for her particular patient. She's saying that if snapshot is negative and ALK is negative, please defer to reflex, ROS, and MET FISH. And this is sort of sad because we're technically rationing out these tests because we just don't have the resources and we don't have the technology to actually keep up with the multiplex nature that is required for you to do a comprehensive type of genotyping for your patient. And this is what we see day in and day out. So more genes, low prevalence, and then the other issue is the tissue. Patient here comes with a resection, bulky tumor, tons of DNA, tons of RNA to work with. Yesterday, this patient drove 2 hours, maybe 4 hours in the Boston snow from New Hampshire, waited another 2 hours for biopsy, and this is all they got. So if you really want to do this at the clinical setting, you want to have success here equally as well as there. And that's the other challenge that we have as a clinical lab. So over the last 5 years or so, we have been offering this test menu in terms of the Center for Integrated Diagnostics, which is essentially our molecular diagnostics lab for cancer. We are essentially a pretty heavy FISH lab, and that's because we haven't found an alternative strategy to be able to replace FISH for copy number and as well as rearrangement detection, not until now. And so our volume is easily 40 to 50% FISH testing. The rest are more one-off types of assay, PCR-based. And finally, what I'm showing you here down here is the new assay that we launched based on the AMP technology as of last August. So in summary, I'm going to stop here and just to say that cancer genetics is quite complex. It's rapidly expanding in terms of the knowledge that we're acquiring. There's clearly demands for molecular genotyping, particularly for oncology, but there are quite a few challenges that I have highlighted. Many targets, limited sample quality and quantity. The turnaround time is key. We're not doing this in a research setting where you have 3 months or so to turn around your results or marinate it and massage it. You really have to get it back to the clinicians within 1 or 2 weeks because the patient is waiting there for you to dictate their management in terms of their treatment. And finally, because of all these issues, this lays the groundwork that we really need higher throughput technologies for cancer genotyping. So if you asked me 3 or 4 years ago at AGBT, are we ready with next-gen sequencing for cancer diagnostics, I would flat out say no. I mean, it's just not feasible, easy to do. It was still expensive. But today, the sequencing cost has dropped so dramatically. The assays are now becoming more readily available. Analysis-wise, in terms of the tools and software, is more mature. So I think today is a very ripe time, and that's why you see a lot of academic centers as well as companies jumping into the game in terms of doing this in the clinical setting. So this is what we thought about 2 years ago when we sat down to think about doing cancer genotyping with next-generation sequencing. You can do the whole gamut in terms of whole genome sequencing, or you can do hotspots and a few number of genes. It runs a gamut in terms of costs, turnaround time, interpretation, the potential for discovery. So we thought that whole exome and whole genome sequencing was too much. For the amount of coverage that we need, likely in the median 500x full coverage or more, these are two technologies that are still quite too expensive for us to achieve that. And again, there's quite so much data that the interpretation becomes the bottleneck in terms of delivering the results back within easily 2 weeks. So we thought in the few hundred gene range, maybe even 1,000, or perhaps hotspots and something dedicated to rearrangement is the way to go. And so therefore, we've selected this strategy, which is a tiered-based testing approach. First tier could be a very small panel, a panel that is kind of like Snapshot, but at the next-gen level, giving you point mutations, copy number, and indel detection so that you can easily get 60 to 70% of the driver mutations that define the tumor. Do that first. If the patient is positive, treat them accordingly. You don't do the comprehensive panel. If they recur in terms of the disease, then you pull up the big guns in terms of the comprehensive panel. For a patient who is negative on these two assays, then you go straight to the comprehensive panel. Or if there's a patient with a dire need and very serious type of condition, maybe you jump straight to the comprehensive panel. So I think this is the correct approach. This assay will be relatively inexpensive, turn around in less than a week or so, whereas this one will take a longer time, cost more money, and demand more interpretation, but that's sort of the only option you have at that point. I think this is the better approach, rather than straight out doing 1,000 genes or the full exome. So target capture technologies have to come into play here if you talk about anything less than a genome or even less than an exome. So these are sort of a survey of what is out there today. You have your hybrid capture, solution phase capture here from Agilent. You have varying, essentially PCR-based technologies on the right side here. But when you look at all of this together, you can note two things. There's really no alternative strategies to target enrichment that affords you the ability to have random start sites. This is highly important, I believe, in terms of next-gen sequencing analysis for variant detection. And secondly, there's really no efficient methods. So let's say you wanted to detect ALK, RET, and ROS, which we know is clearly important today. there's none out there that I think that can do this very efficiently at the next-gen level. So we sought to address this, and that's when we came up with anchored multiplex PCR. Anchored PCR is not a new term whatsoever. It's actually old and dated, maybe 15 years old, 20 years old. But the ability to do this in a multiplex manner, highly parallel and by next-gen sequencing, is something that's quite novel, and that's what we've tried to come up with. So you start first with double-stranded DNA. It could be cDNA, genomic DNA, and then you do your typical library construction in terms of in repair and add A. But when you ligate on your adapter, it's a different adapter. It's not your fully competent adapter. So for example, if you take the Illumina adapter, you have a P5 and P7. Here, the P5 is missing. So while you generate this library and push it through bridge amplification, or if you do an iron torrent version and push it through motion PCR, it's defunct. So it won't generate any bridge amplification or any clonal clusters in terms of your beads. The way we add on the second adapter is by 2 rounds of hemi-nested PCR. The first round has a gene-specific primer here, and using a common primer to the adapter that's on all ends due to the ligation afforded by this step here, you're able to first generate roughly 50 to 60% specificity. You clean that up, come back with a hemi-nested PCR where you use yet another common adapter primer here, but this primer now is 3' of the first one. So in essence, between these 2 primers, you cover about 50 to 60% specificity in the genome, which is quite high, and that affords you about 90% specificity in this assay. In the end, you essentially get your second adapter on, and you can either proceed with iron torrent sequencing or MiSeq or even high-seq sequencing. So let's talk a little bit about random start sites. This is hybrid capture, or it could be whole genome sequencing. You randomly fragment your DNA, you ligate on your right and left adapter, and you are able to produce this nice kind of Gaussian almost like profile in terms of read coverage. If you talk about any other PCR technology, whether it's PCR, Raindance, Fluidigm, molecular inversion probe, HaloPlex, AmpliSeq, or TruSeq Amplicon, they're all essentially blocky types of coverage like this. You need a right and a left probe, a right and a left adapter to dock down to your target sequence, flanking what you are interested in, and then you proceed, and finally you get this type of readout. So whether you have a complex library or input DNA or something that's really, really insufficient and crappy in terms of FFPE, you may not be able to distinguish this at all at the very end of the day when you go to demultiplex, because if you demultiplex this, you lose everything. So anchored multiplex PCR actually gives you a trade-off or a compromise between these 2 scenarios. It'll give you one end that's randomly ligated and the other one that's blocky because that's where your GSP2 docks down. And in fact, you can do this on both the positive strand and the negative strand. And we thought, oh, you have to do that in 2 separate reactions because it would actually cause PCR, right? But we actually just played around, dumped it together into one reaction, and it works just fine. So from a 1-tube reaction, you're able to sense the positive strand and a negative strand and essentially get back this profile, which is very similar to what you get by a hybrid capture or whole genome sequencing. And this allows you to get more accurate sequencing and likely more accurate variant detection and calling. And you can actually demultiplex with routine analysis tools. What is the workflow? This is the traditional MGH protocol. It usually takes about 8 hours or more. The cycling conditions are much longer because we use 5 minutes to help balance out the GC content and bias. But since Enzymatics acquired us, they have tremendously made this process a lot leaner. And they've actually improved it quite a bit in the form of a lyophilized kit. So I run a CLIA lab, and I can tell you this is very, very important because no more do you need to rely on -20 or -80 to store your reagents. Now you can just store this at at 4 degrees and be able to maintain stability for 2 years or even more. This is a set of 7 strip tubes that you literally add on your liquid nucleic acid, put it in, seal it, put it in thermocycler. No more do you need to make a master mix, pull out your primers and pull out your reagents and your enzymes, and the potential for contamination and all of that is gone because it's all dried down. And the protocol now is quite quick. I think this can be achieved in probably less than 6 hours or so in a working day. And this is for RNA-based assay. If you were to do a DNA-based assay, you can get rid of the first 3 steps. And what is not very apparent at this meeting is that Enzymatics also has now an enzymatic shearing solution that is actually combined with in-repair and add A in 1 step. So within 10 to 20 minutes, you'll get your sheared DNA, and you can proceed straight into adaptive ligation, no cleanup in between, into PCR1, and then finally into PCR2. I think this can be done easily in 3 hours or less. So let's go back to mutations and how do we apply the anchored multiplex PCR technology. So I mentioned copy number is important, indels and SNVs are important, but let's focus first on rearrangements and how we are able to tackle that. So currently right now and traditionally, there are a couple of methods that you can use. You can use immunohistochemistry. So if the fusion somehow leads to overexpression of the oncogene or the oncoprotein, and if the background is negative, then you can rely on an availability of some antibody and be able to detect it by immunohistochemistry. However, this is a very wishy-washy assay because it's a pathologist who's reading this, and at the end of the day, they've looked at 300 cases, and they're tired, and they want to go home, and they'd have to say, this is 1+, 2+, 3+, or 4+. So it's not very objective. The second method is FISH, which is very good. It's much more objective than this, but then there's still some subjectivity. Because here, this is an ALK example. That, I think, is a split. This, if you give it to 2 different pathologists, someone may call this a split, someone may call that not a split. So there's still some subjectiveness to the analysis of FISH. It's also technically challenging, laborious in terms of preview. At MGH, every single case gets read by John or myself or Miguel, one of the pathologists, before it gets signed out. And that's a lot of time input. And it's poorly scalable. If you had to do 10 ALK, RET, and ROS, you have to do it on each slide. And now if you have 10 cases, that becomes 30. If you have 20, that's 60 cases that we have to go through. And that's quite a lot of volume for a diagnostics lab. RT-PCR works well, it's cheap, quite robust, can work down to very low input amounts. However, when you start dealing with something like ROS here, where you have 9 or 10 different fusion partners, each one of which may have a different exon involved, you will have to design a right and left primer for every single scenario and be able to do that in singleplex mode. And that can be very time-consuming and laborious. Then you have this other scenario, complex rearrangements, where it's crazy inversions here and there that by FISH, forget it. You're not going to be able to detect, design a FISH assay for. And then EGFR V3 is another great example, something that is a splice variant, and which Bob Daper actually will cover today in full depth. So let's talk about next-gen sequencing. So let's not use IHC, let's not use FISH or RT-PCR, but use next-gen sequencing. Certainly you can do it at a whole genome level. Like the price is much cheaper now than 10,000, but it's very complex, low coverage. So for tumor cellularity at 10% or 20%, forget about 30x coverage and being able to detect that. Transcriptome sequencing is another way to do this, but depending on the expression level of your oncogene or the fusion gene, you may or may not be able to detect, say, 5 reads or 2 reads that cover your translocation. Hybrid capture works well. This is something Foundation Medicine uses, both on the DNA side and also on the RNA side. My opinion is that using it on the intron side in terms of DNA is quite expensive. You're tiling across tens of thousands of bases just to hope and pray that you captured that breakpoint region. And as well, the analysis can be quite complex because introns typically are spattered with repeats, satellite regions that may be difficult to map back uniquely to the genome. So I think it's like putting a sledgehammer to a pin nail here. If you're trying to use these approaches, especially if you know ALK, RET, and ROS is all you care about, then we need a much more focused assay. So in terms of using AMP technology to do this, we design primers that flank the breakpoint region. So let's say the breakpoint is here. This is your unknown gene, and this is the ROS gene that is involved in the translocation. You design multiple primer sets on the 3' end covering 5 exons here, and we also designed 3 or 4 more additional primers on the other end. So differentially, when you go to look at read coverage, you can actually see a differential expression of the 3' end versus 5' end, which is yet another level of evidence to show mutation or actually the rearrangement. So this is some examples of what we have actually detected using this technology on formalin-fixed paraffin-embedded tissue. There's no cell lines here, no fresh tissue. So these are clinical-grade formalin-fixed paraffin-embedded tissue. We've been able to detect all versions of ROS1, even the novel ones that have been reported in the last year or two in Nature Medicine. Most of these are 3' fusions in terms of ROS, RET, and ALK. There's one example here which is the opposite. It's a 5' fusion. So this assay works both on 3' fusions and 5' fusions, completely dependent on how you design your primers and what direction you are marching towards. So here's the anchored in terms of a pileup. You may recognize this probably looks like an iron torrent sequencing because of some of the indels here, but it doesn't matter. In rearrangement detection, you just need to know this maps to ROS and this maps to CD74 or SLC34A2. And here's the randomly ligated in, which affords you unique calling in terms of random start sites, and that's highly important for a confident call in fusion detection. And this example here also shows you splice variant. You see here an exon 34 combined with exon 6 of CD74, here involving exon 35. These are two novel mutation rearrangements that we were able to detect in GBM, NTRK fusions, and we got scooped because it was just published in the last year as well, and we literally detected it right at the same time the publication came out. So we went on to validate this assay on the MySeq instrument, and we took almost 300 cases, formalin-fixed paraffin embedded tissue cases that we had a golden standard answer for in terms of owlfish, rossfish, and redfish. This panel actually also contains NTRK1 and PPR gamma because these three genes are also very important for thyroid carcinoma. So this is a combined lung and thyroid panel. So you can see that our sensitivity is excellent. It's 100%. So in our paper, we had to give some confidence interval, and so it was like 95 or 97 to 100% in terms of the confidence interval. But what you see here are some discordant or actually kind of weird, interesting cases. These cases are ones that we called indeterminate by fish because they are strange and we didn't know what to call. But on the AMPS assay, because of the specificity afforded by sequencing, we were able to definitively call them negative or positive. And I'll show you those in just a few minutes. So I'm showing you here, quite confusing. It's not gene expression mapping here, but rather clinical cases that we've run. So let me go through this. We've run 235 total cases. We've had 15 failures, which is about 6%, and 13 positive. And these are the flavors that we've detected. So what is shown here is each row is a sample. The first column is nanodrop quantitation. Second column is qubit quantitation for RNA. Third column here is RT-PCR, a GAPDH Roche relative qPCR assay where we just giving you CT values here. This is the library yield, and finally whether it passed or is negative in light green, failed is in orange here, and positive detection is in green. So this is color-coded in terms of bad being red and green being good or excellent. So what I want to show you here is that we had a difficult time very early on in terms of failures, and that was explained by a number of things. We had poor amounts going into the assay, as well as our spike cleanup automation system was not set up correctly. We were letting the ethanol sit for like 5-10 minutes, so not bad, not good, right? So we were losing library because of that. So after that, we've changed. The last 150 cases or so have been only 1 or 2 failures. But the failures are not predictable by neither the nanodrop numbers or the qubit numbers for RNA, but rather by the qPCR numbers. You can see some cases are red here, but they turn out fine in terms of working at the end of the day. And so if you look at the the CT numbers from the qPCR assay here, it is highly predictive of failure, and that's what we've been going on to predict failure. And if it predicts failure, we reflex right away to FISH. That way we don't lose any time in terms of getting an answer for the patient. So these are clinical cases that we've run, but to fill out the entire MySeq run, we've also supplemented with essentially research cases. We just threw everything at it. GBMs, thyroid, colon, lung, what have you, what's remaining. And these are sort of similar kind of results that you see here. So the failures were very early on, but since then we have much fewer failures here. And these are the types of mutations that we've detected, or translocations that we've detected. And this is over 6 months worth of work. So I'm going to go into some interesting cases just to show you the power of this technology when you have high-resolution sequence-based detection, how it compares to FISH. So this case was indeterminate by FISH because this has a tandem copy of ROS1. One is normal, and the other one is a tiny red, mostly red dot with a tiny green dot right next to it. So you can see it over and over again. I couldn't call this positive because ROS1 is truly an interchromosomal rearrangement. So typically you would expect a red-green split that's variably far apart in terms of a FISH assay. But I couldn't call this positive, so I had to call it indeterminate. We ran it through the AMP assay, and it was easily positive for CD74 ROS translocation. So the patient should be put on crizotinib. Next case is a green-only out FISH. In the beginning, at MGH, we were the only ones running FISH. And when we saw this, we didn't know what to say. We didn't really have good confirmatory methods. We tried IC, which sometimes was positive and sometimes was negative, but running this through AMP was clearly negative. It doesn't have the canonical EML4 ALK rearrangement. This is a case that was found positive just this Monday. It's KIF5B RET. We tried to confirm by FISH, but it was negative. 2 pathologists looked at it, and I just looked at it myself. Maybe you can call this a split, maybe, but Again, very subjective call there. So we think this is FISH negative. And the reason why is this is a pericentric inversion involving KIF5B and also RET. And they're about 10 megabases apart. In ALK translocation, you're about 13 megabases apart, and you barely are able to detect a split. So 10 megabase may be too small, or perhaps the centromeric region here is just more tightly organized than it is on a paracentric version, which is ALK. And that's why it's negative by FISH. By the AMP assay, clearly positive. So this patient likely should be put on a RET inhibitor. Another very interesting case. This one is a lung adenocarcinoma case, which was RET positive by AMP for TRIM24 RET. We did FISH and saw green-only signals here. Green is appropriate because that defines the 3' end of RET, which is the receptor tyrosine kinase domain. This patient has a long-standing history of papillary thyroid carcinoma, and it just so happens in this same biopsy, there was a focus of thyroid tissue proven by IHC staining. We did the FISH in this one spot here, and you can see it is a red-green split, completely different from the lung cancer portion. How unlucky can this patient be to have 2 RET translocations in 2 primaries? So this is something very interesting that we will follow up by acquiring the primary tumor in terms of thyroid and running it through the AMP assay. So now with specificity of sequencing, we can say, is it the same TRIM24 RET, or is it maybe PTC3 or PTC6? And that can be resolved because we have high resolution with this assay. And just to briefly go through the last few minutes here to show you what is achievable when you sign out a case like this. This is the TRIM24 RET case. We have in there 3 housekeeping genes, GAPDH, TCPP1, and beta-2 microglobulin. Highly important in terms of clinical diagnostics. Because the worst thing you can do when you diagnose a case is make a false negative. And because this is RNA-based and we're working off of FFPE, we need these reads because it tells you that the integrity of RNA is sufficient, and therefore you can confidently sign it out as negative. So what's shown here is black are things that we perceive as genomic DNA if they are intra-exon or jumps from an intron to an exon. Red reads colored here are reads that jump from exon to exon, so we can safely say that they are likely RNA. So you can see there's smooth distribution, high complexity, thousands of reads here, and you can see that it's robust in terms of housekeeping genes expression. This is what you see for the test genes. So this is ALK. There's some very blocky reads here which look like RNA, but I don't believe it at all because ALK typically is not expressed in lung, and that's appropriate. ROS1, RET, PPR gamma, and NTRK1 are all expressed. You can see thousands and hundreds of thousands of reads here, and that's appropriate because we see that all the time. This goes to show you that none of these targets will be applicable for IHC staining because they're all expressed in lung and normal pneumocytes. But if you look at RET exon 12, you'll see that the anchored portion here goes from exon 12 and jumps to TRIM24 exon, I can't read it, but you can see the point there. And you can see that trailing random in. So not high complexity, but you have thousands of reads here, and easily this is a positive case. And of course, we have orthogonal confirmation with the FISH assay. So I love the assay because now instead of spending 5, 10, 20 minutes to sign out per FISH case, I can literally sign a negative case like this out in less than a minute because I just look at this. Great quality, great complexity, high expression of housekeeping genes. I don't detect any fusions, and now I can move on and sign it out as negative. So I love that assay. But now, what about genotyping at the DNA level? So this is a hotspot plus panel that we've developed. 190 amplicons covered bidirectionally, so technically 380 primer sets in one tube. It covers hotspots on the snapshot assay and more. So DNMT3A, the hTERT promoter is all in there. All the new stuff are in there. And we also cover TP53, P10, and CDKN2A, tumor suppressor genes in full. So the entire coding region is covered there. 1,000x median coverage is what we want, using about 400 to 500 megabases of data per tumor. We want about 1 to 2% sensitivity in terms of analysis, and about a 1 week or even less turnaround time if we actually applied the lyophilized kit. The desire is for SNV, indel, and copy number. So here's an example of this assay with an indel. This is your canonical in-frame exon 19 deletion, which would, this patient would be sensitive to EGFR inhibition. This is copy number. I'm taking 48 FFPE cases, tumor, taking the log2 ratio of the normalized coverage against the median across all samples. And what you can see here is EGFR easily pops out for 1 case, 2 case, and 3 case. PDFTO alpha, low-level amplifications. You can see that most of the reads are hovering around here. This is clearly different. ERB2, TP53, GNAS. So this is nothing special here. This is me doing Excel with this data set, and you're able to appreciate some of this copy number change. And here's CDKN2A loss, along with P10 in a GBM case, and STK11 loss in the lung adenocarcinoma case. This is just to show you the concordance between SNV detection and indels across the different samples. And you can see we're able to pick up everything. This is the prevalence or number that we were testing. And if it's detected, it's just no highlighting. We missed two things here, and those are focal amplification. So if you did FISH on these, you'll see a small focus of cells that show the amplification. So it all gets diluted when you do DNA extraction. And I think that's fair to say why it was not detected by AMP. But then we also get these additional calls. So just with this 190 exon assay, we're able to increase the level of detection compared to what we traditionally have done with snapshot. So that 6% likely can be 70% just by doing this one particular assay. So in summary, AMP is a multiplex PCR assay, a target enrichment assay for next generation sequencing. It's so far been robust for gene fusion detection, SNV, indels, and copy number. Economical, I think, in terms of designing it. It's rapid and it's scalable. And there are many applications. So I've already told you about genotyping. Bob Daber is going to talk to you more about allele counting and expression counting. And there's also confirmation sequencing. So let's say you do a big whole genome study and a whole exome study. You have 50 variants that you have to confirm for SNVs, indels, or even copy number. So I think in 2 or 3 weeks, Enzymatics is launching a website that you can actually design your own custom content. Design the primers, and within 3 weeks, you'll get those primers and run it. So instead of running 50, or actually 50 times 2, right, because you do 4 in reverse, Sanger sequence reaction, you can do this all in one tube in less than a working day in terms of library prep, followed by 24 or 27 hours of MySeq sequencing. So I think this is a great technology. Obviously, I'm biased because I developed it, but it couldn't have been done without the work of folks in our pathology lab, particularly John, who is a co-founder and a great mentor in terms of us working together. The Cancer Center and obviously the Enzymatics. I have to say, honestly, we would be nowhere near we are today if we hadn't gotten acquired. Lyophilization, the improvements, the optimization, and just the channels to get this to you, I think we would not be here without them. And so I appreciate all the hard work from top down, and in particular the Boulder group, who is the Archer group. And they've taken something that we essentially jerry-rigged in our garage lab and really made it robust and truly applicable, and hopefully could deliver to you if you have interest in the technology. So I'll stop there for a few questions, and I'll try to move quickly on to the next speaker.